I first started working with field applications of mycorrhiza in the late 70s, early 80s, and we'd have these normally growing trees and these stunted trees. So we would do foliar samples on these plants, and we would notice that these stunted trees were phosphorus deficient. And so the obvious thing would be to check the soil phosphorus levels. Well, it turns out the phosphorus levels in the soil were high, but the trees were phosphorus deficient. And that was because the methyl bromide used to fumigate the soils for diseases also killed the good guys, which are the mycorrhizal fungi. And no amount of phosphorus added to these soils would get them to grow normally. So I was perplexed, as I am often. And I called a professor, Jim Traffy, and he was been a professor so long before they invented hair. But Jim's still actually practicing science. He's still a great scientist. He's still publishing like crazy. And he's in his 80s. But anyway, Jim said, oh yeah, you go out in the forest and you collect these puffballs and truffles and put them in a blender and dilute them with water and spray them over the beds and they'll grow fine. And I thought, how kooky is this old guy? It just didn't seem logical to me because it's nothing I had ever learned anything about. And it worked. And I was hooked, completely hooked. Ended up going back and getting my PhD at Oregon State. Worked with Jim for decades, him and some of his. For those of you that have looked on the microscope and seen the threads, I mean, here's the root right here. These are grown intact in a glass box. But here's the root. I mean, this plant would only have that much root system to here without this fungal colonization. And the mycorrhizal fungus is, I mean, you can have 10 miles of fungal threads in a handful of soil. So it can be profound the way it changes the ability of plants to utilize soils. And they love carbon. They love to get into these carbon substrates. In this case, it was just a mistake. We accidentally dropped some needles into the box. But we actually were able to track the movement of nitrogen out of these needles and into the plant by radioactive tracing of the nitrogen. And we actually learned something from this experiment, that we were actually getting the nitrogen out of the detritus. And then since then, Ecology published an article with our Myco inoculum. I think Tom's going to show another slide of this, but this is a fungal hyphae, and it was published in Ecology, and they actually lit up the amino acids. So you can see the transport of the amino acids into the fungus, and then went up into the roots, and then up into the plant leaves. So it was a big deal because it was the first time anybody had documented the uptake of nitrogen as an amino acid by a plant, which a lot of the old soil scientists from the 1800s are probably rolling over in their graves, because there's always been this thought that nitrogen has to be converted to nitrates to be absorbed by plants. And over the years we've seen, first of all, you can't grow tomatoes without fertilizer. So this is no mycorrhiza, no fertilizer, fertilizer only. And this is a combination of both fertilizer and mycorrhizae. So we're just capturing more of the fertilizer so it's not lost from the pot. Bent grass, this actually might have been before Pennington, but we actually did a study where this is no mycorrhiza, no fertilizer, mycorrhiza only, fertilizer only, with some pythium. And then the combination of the two. So again, better utilization of the fertilizer that we added. And they do this, again, by expanding the root system, but also chemically. These are the mycorrhizal colonies, and they're in plates of insoluble phosphorus. And these halos are where the mycorrhizal colonies are producing an enzyme called phosphatase, which dissolves the phosphorus. So it's an enzyme reaction with the phosphorus to unlock the phosphorus that makes it available to plants. And there's probably 5,000 studies of mycorrhiza and phosphorus uptake. I mean, it's the best documented aspect of the mycorrhizal relationship. And as you know, and probably a lot better than I do, you know, phosphorus doesn't move very well in the soil. So if you have a non-mycorrhizal root, you have this narrow zone of mycorrhizal depletion where it can be absorbed by the plant. And you've got pools of phosphorus that are unavailable. And if you add some mycorrhiza to the system, you can get out into these pools of phosphorus, use the enzyme phosphatase, dissolve the phosphorus, and then transport it back to the plant root. big advantage in terms of access to phosphorus in the soil. And as you all know, good soils have a lot of pore space and big pores that allow oxygen to get in the soil, and medium-sized pores that hold on to water, etc. Well, there's an organic glue called glomalin that the mycorrhiza is a sticky glycoprotein that the mycorrhiza produce that sticks sand grains together. And that's a big part of what soil structure is being able to create structure of these soil particles. And it's a third of the world's soil carbon. So this is a huge reservoir of carbon in the soil and really an important mechanism for depositing carbon in the soil. I think when this is all said and done, and all this mycorrhizal research and all this practical applications done, there may be more use of mycorrhizal fungi for their ability not to improve plant growth, but to store carbon in the soil. Because it can happen fast. 1,000 pounds of carbon per acre per year can be stored in the soil via this glomalin interaction. And you start multiplying that by 200 million acres of farmland, it could be this incredible sink of carbon in the atmosphere. And it also improves soil conditions. The sideline benefit of that is, as some of the other speakers will talk about, the more carbon you can put in the soil, the more healthy the soil is. This is the Rodale Institute, side-by-side plots, high levels of mycorrhizal activity, low or no levels of mycorrhizal activity, add water, and you can see where the silt is catching and floating away. And over the years, we've seen mycorrhizal inoculants really build root systems. In this case, the system on the left. I mean, we can go on and on with pictures of healthy roots. And here's the key. This is a non-mycorrhizal root system. And only the tips of the roots have membranes which allow nutrients to flow into this particular non-mycorrhizal plant root system. If you take one of these roots and you colonize it with, in this case, with an ectomycorrhizal fungus, you get this like coral reef effect of mycorrhizal activity. And now you've got all of these absorbing surfaces. This whole structure can absorb nutrients and water out of the soil. So you've just, again, increased the amount of absorbing area. And we found, and I think people, again, you guys should be experts on this one, but especially this year, we found in the drought in the Midwest that better roots mean better yields. This is treated and untreated plants. Way back in 2004, in Fort Myers, Florida, we had a Hurricane Charlie hit, canceled the flights, stuck there, but of course had mycorrhizae on us. So we actually, this is kind of a local tree, and we actually inoculated the root system. You know, a few days later, the tree was re-erected. This is what the tree looked like 3 months later. And this is what the tree looked like 13 months later. So root growth is...
How much of that is stored nutrients?
In this particular tree, probably somewhat. But a tree that size needs feeder roots. And the ability of the mycorrhiza to produce tremendous proliferation of feeder roots. We had access to the grass. This is a classic study. I've showed a lot of this picture to a lot of you, I think, in the past. But these are treated corn, subjected to drought, and you can see the untreated corn wilting. Well, what's really fascinating about this picture is you can see where the mycorrhiza is spread across the controls and started to colonize the basin area at the University of Florida. And then just 2 weeks ago, we discovered that the University of Wisconsin had used our product on another corn study, and they had 28% longer survival of the corn, of the inoculated corn, compared to the control corn. So this was just published a couple weeks ago, and it used our Myco endo product. So we feel like it's a real tool for managers in low water conditions to improve growth and develop drought tolerance. And it's done in a variety of ways. I mean, one of the ways is these fungal threads have much less diameter than the roots themselves. And so they can get into the fine nooks and crannies of the soil to absorb water. And it's the fine nooks and crannies of the soil that hold water when water becomes limiting. And the ability of these vesicles to store water is also a huge feature to absorb water and then slow release it to the plant. A new aspect of, you see a lot of new studies coming out all over the world about weed control with mycorrhiza, organic flax producer. And we've heard from many of our customers over the years that the most profound difference that they noticed from using the mycorrhiza is less weeds. We actually got a picture from one of Gary's customers just a couple weeks ago, side-by-side trial, and far less weeds in the treated compared to the control. And right now the literature is showing that the reason why weeds are getting outcompeted in these mycorrhizal treated beds is that they're starved for phosphorus. In a very disturbed non-mycorrhizal environment, these weeds are very good at getting phosphorus out of the soil, better than the mycorrhizal plants because there's no mycorrhizal present. But you put a healthy mycorrhizal colony in there, the weeds get outcompeted because they can't get enough phosphorus for their own growth. So it's probably a P or a P-related factor. We're very, this company has spent a lot of time encouraging people to research our products, and we've got 23 published studies on our Myco product. Most of that's happened in the last 3 or 4 years. And so it's been interesting to look at some of the study plots. This is at the University of California, Riverside. You can see the mycorrhizal treated plots just in terms of grow-in. I think some dog or cat could be doing it. I'm not quite sure what that is, but so we get very good results in terms of cover and root growth, and that was actually published in Turfgrass Science. And here's the one that just recently come in. It's actually from last year, but we just got the data. But these are potatoes, inoculated versus non-inoculated, and this is the revenue per acre. So there was over $1,000 increase in potato revenue per acre by adding the mycorrhiza. And it's interesting, the best yields were actually where we had reduced the phosphorus levels from 120 pounds per acre to 30 pounds per acre. So again, that's sort of that improved phosphorus efficiency. This plot probably had tons of phosphorus in the soil, but it wasn't available until we added the mycorrhiza and it suddenly became available. So that probably is one of the reasons why they got such a high net revenue increase. And then over the years, people have compared mycorrhizal inoculants. This is Tarbell and Koski from the University of Rhode Island, and this is our inoculant. They compared 8 different inoculants and 3 different rates, and so that was our product. We were very proud of that one. This is the University of California, and looked at shoot height, leaf area, and total drive mass. And again, here's our product, which did very well compared to the other products. Comparison of 7 products in terms of mycorrhizal colonization. This is our product. We got the best mycorrhizal colonization at the University of California. This is Purdue University. This was our product on, I think it was onions and watermelon. And this is the amount of mycorrhizal colonization. We had the best product. Here's the control here. So, I mean, another study, our granular product and our micronized study for commercial products. This was published by Myco et al. So we really feel like in comparisons of inoculum, you know, our product is faring well compared to other inoculums. And I'll try and explain to you why here in a second. There's some features of our inoculum that we think that make them superior. So how and when to use the mycorrhizal inoculant? We're going to get a lot of feedback from you on this. What's the best way to use the inoculant? This is probably not the best way to use a stick and pour it in the seed box, but it did work. You know, that's what he happened to have. But here's our powder. They're actually mixing it with wheat seed in a seed box. But, you know, we have various forms of inoculum. We have our granular form, which can be banded with seed or broadcast. One of our more popular emerging products is our liquids. And many of you are starting to use that now, and we think that's a major advancement in terms of applying mycorrhiza in the field. I mean, there's a lot of ways to get it on the seed. You can pre-apply it at the seed company. You can mix mechanically at the farm. You can ban it with seed at sowing. So there's all kinds of ways to get it with seed, and getting it on early is very important. And you can, I mean, for the grape industry, you could dip cuttings, rooted cuttings into mycorrhizal slurries, or like for forestry or horticulture, you can actually inject spores through these sprinkler systems, and you could inoculate 1 million plants in 7 to 10 minutes just by injecting the spores, and they come in and they rain through the sprinkler systems into the soils. And this is actually how we started as a company. So what to look for in products, and I think this might be help explain why our products are doing better, because we've learned through trial and error that the best products are concentrated. And also make sure that you look for a label with the product. If somebody's providing you mycorrhiza without a label, avoid it, because that tells you that they really probably don't know what's in it, or what species are there, or what concentrations. So make sure there's a label there. Multiple species, different mycorrhiza do different things for the plant, and so that diversity really relates to better performance. And we actually selected our fungi for their performance. A lot of mycorrhiza are totally intolerant to fertilizer. So when we start growing mycorrhiza, if it was intolerant to fertilizer in the early days, we didn't grow it, because fertilizers are a fact of life. And so we select our fungi for their ability to improve plant growth. And we've dumped several strains and species because of that. It's obviously got to be cost effective. And again, we're looking for your feedback on that one. It's got to make sense economically. And you need good advice. This is some of my favorite advice. We have the best registration people in the industry. Has everybody met Christina here?
Yes.
Christina in the back, and Tanya. Is Tanya back here?
Oh, she stepped out.
Okay. And Jeannie helps out, but I tell you, you do not want to get into this regulatory environment without support, because it's very important. And we're very proud of the people that provide support and advice to people about getting products registered and kinds of claims they can make, etc. This is also important advice. It's amazing how much common sense we have to circumvent in the registration process, but we have to label everything and be very clear about what we're doing. Do not drink this water. So we talked about diversity and why it's important, and there's obviously some very important reasons. Is that some are better at phosphorus uptake, some are better at drought protection, disease protection. Some produce a lot of enzymes, some appear to be better at producing glomalin in the soil. And there's so much range in plant, soil, and climatic conditions. You really need diversity. So the plant will choose the mycorrhizal fungi that are going to give its best benefit in that particular environment. We really believe in the science. This is what's going to drive the future of this whole product. And this whole mycorrhizal inoculum is the science behind it. And so we continue to work with the universities. When the universities do studies, they usually come to us for inoculum. So that's kind of nice that they recognize that we have the quality inoculum. We're really building on the work that some great scientists have done starting in the 1950s. So, a lot of this we're sort of reinventing the wheel, but we're putting it to practical application. And we need your help. I learned so much even from last night, just from talking to you guys about what are some of your issues, your challenges, the opportunities that you have with biological inoculants. So I'm looking forward today from hearing from you and seeing what the future is going to look like in terms of mycorrhizal inoculant. And last night I sort of talked about that Leonardo quote about, Leonardo said that we know more about the celestial movements of the universe than we know about the soil right beneath our feet. And so here's the soil universe. And I feel like the Star Wars theme should be playing now or something. So at the end of the day, hopefully we can prove Leonardo wrong and we'll learn a lot about this underground universe that helps support plant growth. So do we have time for a few questions?
We do.
Very good. You want to turn on the lights back there? Is anybody awake? We'll do that pretty fast. Yes?
Mike, I was wondering if you had any studies that estimate the amount of global warming or air change that has occurred because of our agricultural practices over the last 150 years, 100 years?
There's a guy at Ohio State that's, I can't remember his name, but he's done a whole set of fabulous studies. He actually has documented carbon dioxide trapped in ice when people first began cultivating soils. Because it goes back not just in the last 100 years, but when cultivation began, they saw a spike in the carbon content of the atmosphere as trapped in these glacial ices from when Mesopotamia was first cultivated. So there's been spikes in the Middle Ages when there was less cultivation, the amount of CO2 in the ice went down. So a lot of the effects of agriculture are related to tilling. So every time you till, you mix oxygen with carbon, and you get a flush of CO2 in the atmosphere. And some countries are considering using, like Australia is considering getting a carbon credit for the use of mycorrhizal fungi. And there are already no-till credits in place where if you get no-till, you get carbon credits for so many metric tons of carbon that you can store in the soil. So it's coming, definitely coming. Yes?
What's the effect of glyphosate on mycorrhiza?
Good question. The earlier research, which was done by glyphosate companies, showed no effect. But some of the new research that's coming out is showing that there is an effect because of the repeated use and the residual glyphosate in the soil. So, for me, the jury's still out on it, but it's getting serious in terms of what glyphosate is doing with repeated application to not only mycorrhiza, but all soil biology. So it's a concern. Good questions. Also, no one brought up fungicides, but we have fungicide lists. Some fungicides adversely affect mycorrhiza, others do not. Some actually encourage mycorrhiza. They did a nice study in biological sciences with our product with, I think, 10 or 12 different seed applied fungicides. And the nice thing about seed is there's such a minute amount of fungicide on the seed, it solubilizes really quickly. And it appears as if mycorrhizal inoculant on seed at the levels that they put it on seed is not having an adverse effect. So the seed fungicides seem to be very compatible with mycorrhizal fungi. And if you are using a drench of fungicides that adversely affect mycorrhiza, you have to really think about the timing of when you get colonization or the timing in which you re-inoculate, because the last thing you want to do is inoculate and then drench in a fungicide that affects mycorrhiza. It's a waste of money, obviously. Yes?
Do you have a list of herbicides too that would be detrimental and some that don't affect them?
Well, now we don't have any herbicides listed, but that could change. That could change. And there's also some insecticides that could have an effect. You know, we're starting to see more in the literature. So we're constantly revising our list to try and keep this updated as possible. So, yes?
I should you say that some fungicides encourage mycorrhiza activity. Is there seed applied ones that do the same thing that you identified?
In the study that they did with our product and all those, I don't think any of them actually encouraged mycorrhizal colonization. Some had slightly higher levels, but it wasn't significant. So, but I know like the phosphite, kinds of fungicides are actually encourage mycorrhizal development. Yes?
Are the mycorrhizae, are they evolving just like all other creatures on the earth?
The ecto are, but the endo, the question was, are the mycorrhiza evolving? And the endo, I feel so bad for them, they haven't had sex in 460 million years. They're asexual spores, and they really haven't changed. I mean, I've seen some great papers where you look at the spores look just like they look when they had the microscope. They haven't changed, they caught them in amber. They just haven't changed genetically or anything. And it's because they're asexual spores and there's not a lot of DNA, but the ectos are changing and evolving with the endos. And there's a lot of specificity in the ecto group. Like there's certain fungi, ectofungi that you wouldn't use on certain trees, but you would on other trees. So there's specificity for the ectos. That's why we put a lot of different ectos into our plants. We're trying to capture oaks and conifers and, ecto hardwoods. And so we put a lot more ectos in because there is specificity. But the endos, it's amazing. I mean, like Lamus interradices, you'll find it in Alaska, Jeff's backyard. Guarantee it's there in Alaska. You find it in Costa Rica. You find it in Siberia. You find it in Spain. You find it in Patagonia. And it's Lamus interradices. It's the same fungus. It's crazy. And it might be on, one plant in Alaska and one plant in Costa Rica. It's totally different kinds of plant groups. But it's just an amazing fungus.
So with heavy tillage and that kind of thing, these structures aren't going to evolve to live in these types of environments, the endos, so much. If something's detrimental to them.
They don't like they don't like unnatural kinds of disturbance. And they're slow to recolonize, at least on our time scale. But they're everywhere, and so they eventually get back to where they need to be. Because they occur on 90% of the plants, and they go from root to root. So they've been probably the most successful organism on Earth, if you think about it. So, but they don't like cultivation, they don't like fallow, they need plants as a partner. Without plants, there's something. These are great questions. Yes, Tom?
I ran across some recent research where mycorrhiza apparently facilitate communication between plants whose roots are interconnected with them. They can actually pass information from plant to plant about an insect attack from some distance away and cause a plant immune response down the line. Are you familiar?
Yeah, Susan, we actually started a lot of that looking at fire recovery and the connection between the plants that sprouted, the hardwoods that sprouted after a forest fire, and the ability to get conifer seedlings to survive after planted. And they tended to survive better around these refugia of sprouting hardwoods, which kept the mycorrhizae alive. So when we planted the trees, they would connect into that network. So you'd have like a Douglas fir and an Arctocephalus bush connected by the same fungus and allowing reestablishment of the trees. And then Susan Smart up in Canada has done all kinds of interesting communications kinds of work. And, again, I read it, but I'm not sure the implications of that for a soybean field or a corn field. But we know the mycorrhiza spreads through a field. We try to get so much out there, and we know it spreads from root to root.
I know there were some studies too about sharing nutrients between plants when they actually cut off one plant from photosynthesis and they blocked it from the sun, and it started drawing nutrients for survival from other plants in the area that weren't cut off.
Well, what I'm suggesting, or what this research is suggesting, is, for instance, if one plant in a community that was interconnected with mycorrhiza gets visited by aphids and has an immune response as a result, before the aphids get to the other plants, through the mycorrhizal connections, those plants are alerted that there's aphids in the area, and they already come up with an immune response.
It is possible, because they are connected and they are exchanging stuff. They're exchanging all kinds of compounds. So nature continues to amaze me. It just continues to amaze me. So I would not be surprised if they're.
And they cut all those plants off from the ability to communicate with volatiles also that the plants are.
Yeah. And we know what happens with volatiles and pheromones, but
they cut that out. They eliminated that. And so mycorrhiza is very the only way that they.
Very interesting. You guys talk to some of your farmers out in the field about that, they'll think you're crazy. But it's interesting. So our next speaker is Jeff Lowenfels, and Jeff is sort of our keynote address here. And I've heard Jeff speak several times. He's fantastic. Do you want to introduce him?
No, you're fine.
Okay.
I was just gonna suggest, we're about 10 minutes ahead of schedule, which is great. We're gonna need that, and when Mike finishes this little introduction, we'll give Jeff a minute to set up. And if people do need to take a quick bathroom break or something, maybe we should.
The bathrooms are, if you go past the tree, the two doors on the left. Don't use the tree. So past.
a lot of country people
I didn't want to cut off the questions if we still have time, so anybody else?
Yes, I just had one more on that. So you said in the study where the conifers, there was high phosphorus in the area, but yet the conifers weren't taking it up. Now, I think I remember someone saying that in high phosphorus environments, sometimes these spores will germinate. Is that correct? Or
It could be the chicken and the egg, and that's very true. It could have been, there was an inoculum in the soil, but the soil phosphorus levels were so high that the spores didn't germinate. And so, well, that's an interesting, I don't think anyone's ever measured that. So that is interesting. And generally for our species, if available phosphorus gets above 70 parts per million, we do see a delay in spore germination. It doesn't kill the spores, but does delay the germination. So it actually could be a factor. 30 years and I never thought of that. I hope you're happy. Anybody else?
Are certain species of mycorrhiza more phosphorus specialists than others? And does the mix of species that you have in your product respond differently to different soil phosphorus levels?
Some species and strains produce more phosphatase enzyme, which is the active part of making phosphorus available. I think that's fairly well documented in the literature. So if we don't get a bunch of phosphorus uptake, we don't want that fungus in the mix. So they all help some, but there are certainly some that have been shown to be better, like Thomas Monsier, for example, is one that's just really prolific at sucking phosphorus out of the soil. Yes?
This shelf life of the dried products versus the liquids and the products I see commercially that are soil bags that say now with mycorrhizae, what do we do for stability so they don't germinate and just fail?
Yeah, it's
Given different moisture contents. Are there
Yeah.
some that are stable in liquid and some that are not? Or
Yeah, this
it's
is-
very confusing to
That's
our
a
end
good,
users.
that's a good follow-up on Paul's question. I think the reason why these organisms have been so successful is they don't germinate every time there's a little heat or moisture in the soil. It takes root activity. And they're gonna need that root to survive. So they don't germinate until there's root activity, and the root's off a chemical messenger, we call it forminonetin. There's a commercial product out called Myconate, which is a salt-based forminonetin. But anyway, it's that compound, there's probably several others, that roots pump into the soil that tell the spores, okay, I'm your energy source, form the relationship. And they'll actually grow right to wherever that enzyme's, whatever that compound is being produced. So I mean, you could actually trick a spore. This is kind of weird, but if you put a living plant next to a petri dish full of spores, all of the spores germinate right towards the root system. And you can flip it on the other side, and it'll turn around and start going to wherever the root activity is. So they're definitely attracted to that root compound. And that's why if they're safe to store in your bags for 2 years, even though there's some moisture in your soils, or even floating around in sterile water, they just float around. But they're not active until there's root activity. Okay, with that, we'll give Jeff a little extra time. I think you're going to enjoy Jeff. And again, this Teaming with Microbes book is available, and the next book will be out in about 6 months. 3 months, 4 months. 4 months. Very good. Be good enough.